Okay, good afternoon, everybody. And I'm sorry that I have to do it this way, but let's get on with our discussion about metabolism. We ended up last class, we discussed enzymes in detail, and we ended up looking at considerations about things that affect the functions of enzymes, such as pH, temperature, etc. Here is another set of constraints upon the action of enzymes. And this is the question of this is the fact that certain substances are actually able to inhibit the activity of enzymes. And the first one that we're going to consider is this here. These are competitive inhibitors. Now, notice that that's really important, okay? These are called competitive inhibitors because they compete for the active site with the normal substrate. So this is the way the competitive inhibitors work. They actually are able to bind to a part of the active site. Remember that the active site has a shape which is exquisitely suited to the shape of the substrate. The substrate has to fit in absolutely to the active site for the enzyme to be able to work, to form that enzyme substrate complex. Competitive inhibitors are able to bind to a part of the active site. And what is really important about them is that they bind irreversibly. Once they're bound there, they stick there, and the substrate cannot fit in to the active site anymore, so the activity of the enzyme stops. Now, this is not a natural way to control enzyme activity in the cell. This is a situation, an artificial situation. This is typical of things like poisons and toxins. For example, the poison cyanide, hydrogen cyanide, for example, binds to one of the respiratory enzymes, to the active site of one of the respiratory enzymes, and stops respiration, aerobic respiration. And that is why cyanide is lethal. It literally causes cells to suffocate from the inside out. But remember, this is a totally artificial. This competitive inhibitor comes from outside, from a source external to the organism that it is poisoning. This is also how some snake venoms work, some spider venoms, those sorts of things. These are competitive inhibitors on enzymes, and they effectively kill the enzyme activity. On the other hand, the next set of inhibitors that we'll talk about are not called the non-competitive inhibitors, and these are one of the most important ways in which the cell naturally controls enzyme activity. So what happens with competitive inhibitors, they bind permanently. Non-competitive inhibitors do not. In fact, it's essential for their functionings that their binding is really temporary. And this is the way that they work. First of all, let's have a look. Here's our enzyme under consideration. There is its active site. But if we look at an enzyme which can be inhibited by a non-competitive inhibitor, you'll see that there is another site here. Now, this kind of inhibition is also called allosteric inhibition. And this place here is the allosteric site. And the allosteric substance, the non-competitive inhibitor, binds there. And when it does so, it alters the shape of the enzyme, and it alters the shape of the active site so that the active site can no longer bind substrate. What is really important about this is that this binding is not permanent. In fact, it's very temporary. This goes in and out. It bounces in and out effectively. And as long as it's bound here, the enzyme is off. When it's away, the enzyme is on. When it's in, it's off. When it's out, it's on. Okay. You'll see the importance of this just in a minute when I explain to you how these actually work in the cell. So remember, this is an allosteric site there. It's binding the non-competitive allosteric inhibitor there. And it's altering, when it's bound like that, it alters the shape of the active site so the substrate can't bind. So how does it work in real life? Well, remember what we said before, that these metabolic systems usually run in a pathway. So for example, here is one. We have an enzyme which is binding a substrate. Here's the substrate. It binds the substrate, and eventually an end product is produced. But the end product is not produced by the first enzyme. The first enzyme produces an intermediate. It alters the substrate some way, produces an intermediate. A second enzyme binds that and creates another intermediate. A third enzyme binds that and ends up producing an end product. This is a metabolic pathway, a typical metabolic pathway. And the complex systems usually run in these metabolic pathways. So notice that this first enzyme here is an allosteric enzyme. There's its allosteric site there. So it offers us the opportunity, we're going to see, to control the activity of this enzyme by allosteric inhibition, by non-competitive inhibition. But what is the allosteric substance that is going to do that? It's very simple. It's the end product. And this is the way it works. Remember what I told you. This binding at the allosteric site is purely temporary. This bounces in and out continuously. When it's in, the enzyme's off. When it's out, the enzyme is on. So just picture this. Let's say here's our first enzyme, right? And there's very little end product being produced by this pathway. If that is the case, then this allosteric site will be bound very, very seldom. Every now and again, one of the end products, will bind there. But it only binds temporarily and it's out, and then the enzyme. What about if you produce a lot of end product? If you produce a lot of end product, if the concentration of end product is high, that allosteric site is going to be occupied more often than not. The enzyme is going to be off more often than not. What does this do for the cell? Well, the higher the concentration of the end product, the less activity we have in the first enzyme. It slows the whole pathway down. If there's lots of end product, we don't need to be producing. So it slows the whole. But this end product, don't forget, is being used up. It's being produced for a reason. It's not being produced so that it switches this off. It's one of its functions is that it acts as a non-competitive inhibitor. But that's not the reason it's being produced so that it can be used for something. And as it's used up, the concentration drops. And as the concentration drops, this first enzyme is on more often than it's off. It's on more often than it's off. The pathway speeds up. The pathways will speed up and produce more. So this is a very, very fine-tuned way of controlling the activity of a whole pathway. And it is called feedback inhibition. Feedback because we have an end product. And this is very irritating. We have an end product. And, sorry, I'm trying to get rid of this. And the end product feeds back to this enzyme here. Okay, it feeds back to this enzyme. Now, why don't we have all of the enzymes being controlled by non-competitive inhibition? You don't have to. All you need to do is control one enzyme, and you control the whole pathway. There's no point in having this one be the allosteric enzyme. Because if you do that and you control with this enzyme here, you produce all these intermediates and it's a waste. The pathway is meant to produce this end product. You control from the first enzyme so that when need be, the whole pathway gets switched off. You don't waste energy and resources producing intermediates that you don't need. This is a very important concept, and it's a way in which these metabolic pathways are widely controlled. And in the vast majority of cases, it is the first enzyme that is the allosteric enzyme. There are one or two pathways where one of the other enzymes might be allosteric as well, but that is usually because the pathways are branched, and the branchings are allosterically controlled as well. But non-competitive feedback inhibition is one of the most important ways of controlling enzyme activity in the cell. I just want to remind you again that the vast majority of enzymes in the cell are protein, usually very complex protein. But there is another substance which can act as an enzyme, and that is RNA. And when RNA functions as an enzyme, we call it a ribozyme. And ribozymes are tremendously important in the cell. First of all, the ribosomal RNA proteins, they are enzymatic, and they are primarily responsible for assembling amino acids into protein at the ribosome. But small ribozymes also operate throughout the cell, and they're involved, for example, in gene control, in trimming messenger RNA. All sorts of functions are accomplished by the ribozymes. They're a very important set of enzymes, even if they only comprise a small proportion of the total number of enzymes in the cell. Okay, before we move on to talking about respiration and all of these complex topics, I just want us to remind ourselves about the nature of the kinds of reactions that we are going to be talking about. Remember that the energy coinage in the cell is the electron. It's electrons with a tremendous amount of energy that are going to be traded in order to produce, ultimately, we're going to see, to produce ATP, biologically useful energy. But let's remind ourselves that the process of removal of an electron from a substance is the process of oxidation. Electrons, however, have to always be donated to a receiver. So there is a donor and a recipient. The donor is oxidized when an electron is removed, and the recipient is reduced. And those reactions are always coupled into an oxidation. Here is the oxidized substrate there. Here is the reduced substrate there. That's a reduction reaction. This is an oxidation reaction, and we refer to the whole thing as a redox reaction. And they are always paired like this. You can't have one without the other, because we can't have electrons just flying free in the cell. That's not the way the physical world works. So we've now clear to us that what we're going to be doing is we're going to be trading electrons. But I've already said, you can't just have electrons just moving willy-nilly from one substance to another in a biological reaction. Instead, the electrons must be temporarily captured by a substance which is reduced, which then moves those electrons to a substance which itself is going to reduce this carrier. And this is the electron carriers are there's several of them within the cell, and I'll remind you in a minute that they very often interact with enzymes. And these electron carriers are reduced by accepting whole hydrogen atoms. Hydrogen atoms simply consists of a proton and an electron. It's the electron's energy that we're particularly interested in, but the protons also become important, as we'll learn in a while. So biological systems, remember, we talk about whole hydrogen atoms being removed. For example, if you look at a fuel substance, which consists of many carbons, all those carbons have associated hydrogens. We're going to watch as those hydrogens are stripped off and the energy of their electrons is harnessed. But they have to be moved from their source. So here, for example, we have a fuel source. It can be glucose or a lipid or whatever it is. You can be sure it consists of many carbons with associated hydrogen atoms. Here's our fuel source. Here's a hydrogen atom there, proton, electron. Here's another one here, proton, electron. What we are particularly interested in is removing the electrons from there, but we can't just grab electrons there. We grab the whole atom. And this here is an electron carrier. In this case, it is NAD+. This is the substance which is in its oxidized state, NAD+. What we're going to see is we're going to remove 2 hydrogens from our fuel source. We're going to oxidize the fuel source, and we're going to reduce NAD+. If we do that, what we end up with, here's the oxidized organic molecule. There, it's lost 2 hydrogens. Those 2 hydrogens now come over to reduce the NAD+. If it's plus, it means it has a positive charge. One of those electrons is going to be equivalent to that positive charge, so it's going to lose its charge, and it's going to accept a whole hydrogen to become NADH there. Here's the electron which neutralizes the positive charge, and we have a free proton. Protons are perfectly at liberty to be free on their own because they go into solution. And remember what I've told you, if you increase the proportion of protons, you create an acidic solution. So all this happened here, we've reduced the NAD+. It's accepted a whole hydrogen and 1 electron, and associated with it is a proton. This now can be physically moved as a carrier. It can be moved to the site of a reaction which requires the energy associated with these electrons. So this is one of our most important electron carriers, but there are several others that we will hear about. So I just want to remind you that you've seen this slide before. Many enzymes consist of several parts. Before they can become active, they have to have all of their parts in play. This is the protein part of an enzyme. It is inactive because it's not complete yet, because it doesn't have its cofactors associated with it. This enzyme requires cofactors in order to become active. In this case, what is going to happen, in this case, what is going to happen is that this portion here is a site where the cofactor, in this case, is a coenzyme because it's an organic cofactor. And the organic cofactor is a substance like NADH that we've just seen. So let's say the NADH, let's call this NADH. It's a coenzyme because it's an organic cofactor. It's going to bind here This purely protein part is the apoenzyme. Now it's the holoenzyme, the entire enzyme. It has its electron carrier bound there. Now its active site is complete and it can bind a substrate. And guess what's going to happen to the substrate? The substrate is going to be reduced. It's going to be, the enzyme is going to facilitate that reduction, because what it does is it brings the substrate into close contact with the coenzyme. And it may, in fact, do a couple of other things chemically to ensure that this substance now is reduced. And what has happened now is that this substance has the energy of those electrons associated with it. Where did the electrons come from? They came from a fuel source, right? They came from the fuel source. We've moved energy. We have physically moved energy now to this substrate. I just mentioned as well that not in the case that we're talking about immediately here, where we have electron carrier, but I'll also point out that this coenzyme often may, depending on the enzyme, what the enzyme is there to do, it may actually be something which is rich in phosphate and is able to transfer a phosphate to a substrate. And that is what we refer to as substrate level phosphorylation. And we'll talk about that in a minute. Just remember that that may also be achieved by a coenzyme that associates itself with a particular enzyme. These are all electron carriers that we're going to encounter. We've already heard about NAD+, comes NADH plus a proton. This one is very similar, nicotinamide adenine dinucleotide phosphate, very, very similar to NAD, excepting that this one is most common in plants and in plant photosynthesis. This one is another one which which are used widely in cells, whether they're plant or animal, and that is FAD, and it is reduced to FADH2. This one here is coenzyme A, and coenzyme A is one of the most important of all of the coenzymes and facilitates many, many different kinds of reactions, as we'll hear later on. Okay, the whole topic of our discussion today is to ask, how is ATP made? How do we take energy, say, from some energy source, either a fuel source or something like that, take that energy and put it into our ultimate biologically useful substance, which is ATP? How is it that the energy of electrons ends up forming that third bond of ATP? Well, there's three ways in which it's done. The first is called substrate level phosphorylation. Substrate level phosphorylation is a relatively simple enzyme facilitated chemical reaction where we take a phosphate source and we take ADP and we add the phosphate source to the ADP and form ATP. We'll see an example in a minute. The second way is that ATP is made in the cell is by oxidative phosphorylation. Now, oxidative phosphorylation only occurs in organisms which utilize oxygen. And we're going to talk at length about how this is done. The reason being that our substrate level phosphorylation reactions, they produce relatively little ATP in the cell. For some organisms, such as anaerobic organisms that don't use or cannot use oxygen, substrate level phosphorylation may be all that they have to produce ATP, and it's enough for them, but it's not very efficient. Oxidative phosphorylation, however, produces abundant ATP compared to substrate level phosphorylation. And this is the primary way in which oxygen utilizing organisms produce their ATP. So the third way is by photophosphorylation, and this is in organisms which can use light as a source of energy. And so it's unsurprising that this, for example, occurs to a certain extent in photosynthesis. It does not produce very much ATP. These systems do not produce very much ATP. They can be harnessed to produce ATP if the cell needs it under special circumstances. But we'll just discuss it very briefly in a minute. So let's have a look first at typical processes of substrate level phosphorylation. And this occurs, for example, when we have a phosphate, a substance which has been phosphorylated, which has a phosphate associated with it. And we bring it into contact with ADP and enzymatically remove this phosphate there and add it to the ADP to form ATP. Here's a typical reaction. So this is some substance. It doesn't, it's phosphorylated substance. And what we're simply going to do is we're going to take this phosphate and we're going to add it to ADP, and we're going to end up producing ATP. We're going to strip the phosphate off there. We end up with a dephosphorylated compound, and we're going to phosphorylate ADP to produce ATP. Here is a very typical reaction, a real reaction that it does occur in what we're going to be talking about in a while, about respiration. But I don't necessarily point it out again because I'll probably forget. But here's a substance, 1,3-bisphosphoglycerate, 2 phosphates. Here they are, here, there, and there. Okay. And what is going to happen is that we're going to strip that phosphate off and we're going to add it to ADP. And this is accomplished by an enzyme, and that is phosphoglycerate kinase. And a kinase is an enzyme which specifically moves phosphates. So this is the responsibility of this enzyme to dephosphorylate the substance and move the phosphate to ADP to create ATP. And we end up with a 3-phosphoglycerate. Here's just 1 phosphate left. That is a typical dephosphorylation of the phosphate source and phosphorylation of ADP. But these systems, as I say, are relatively unimportant sources of ATP in the cell where aerobic or oxidative phosphorylation is possible. And we're going to spend a lot of time talking about oxidative phosphorylation. So don't freak out about these diagrams. You'll understand more later on, because you are going to have an obligatory extra credit exercise on photosynthesis, and this will become more apparent to you. I'll use this first of all, this diagram. This is actually a diagram of plant photosynthesis. And you probably covered this in biology, and you'll remember that there are 2 photosystems in the cell which are responsible for harnessing the energy of light and exciting electrons, and the electrons are passed through electron transport chains and end up producing energy-rich compounds. One type of energy-rich compound that they produce is ATP, but it produces relatively small amounts of ATP. Much, much more important in photosynthesis is the production of NADPH. And if you'll go back a couple of slides, you'll remember that I told you that NADPH is an electron carrier. And all of this energy-rich stuff, the NADPH electron carrier, the ATP, this all gets used up in the process of fixing carbon dioxide into sugars. So that is the purpose of the production of these energy-rich compounds, is so that it can be used in a very energy-demanding process of turning simple carbon dioxide and water into energy-rich sugars. This, if the plant cell, or the cell rather, the cell that is photosynthesizing, performing plant photosynthesis, needs to do so, if they need a source of ATP for other metabolic processes, they are able to separate off this part of the photosynthetic process just to produce ATP. So this is usually a temporary situation, and they can actually harness a cyclic system in order to produce lots of ATP if it happens that they need it. But don't forget that plants, in addition to photosynthesizing, plants actually simultaneously utilize oxidative phosphorylation or aerobic respiration. So the two processes run in parallel. Okay, so what we're going to do now is we're going to start talking about the metabolic pathways that will produce useful biological energy in the cell. And these are produced by complex metabolic pathways. And they are what we're going to watch is how we extract energy from organic compounds such as fuels. And we're going to end up putting it into ATP in order to fund those very expensive, energetically expensive biological reactions that create biological molecules, for example. Remembering that all of the activity of these pathways is directed by enzymes. Very few of these reactions happen spontaneously. Instead, the rate needs to be increased by the use of enzymes. So I'm going to stop there for the moment, as like class 1, if you like, and take a little break, and then we'll return. Okay, so back to the drawing board. What we're going to talk about now is how the cell actually acquires energy from carbohydrates. So we consider carbohydrates because it's relatively simple to imagine how we can abstract whole hydrogens from a compound and then start moving them through whatever processes we need in order to acquire the energy of associated electrons. But remember that in actual fact, we can use other substances in the cell as a fuel source. And that includes things like lipids, which are a common fuel source in the cell, fats, even in dire circumstances, you can even start to use proteins. And that is an unusual situation and usually the result of starvation of the cell, that it will begin to use up its own protein stores as a fuel source, all in a desperate attempt to feed the metabolic pathways with ATP. So we're going to now consider we start off by considering just the relatively simple molecule of glucose. And most of our discussion will be how much energy can we actually extract from a molecule of glucose. So we need to remember that in microbes, in microorganisms, there may be two entirely different systems of producing ATP. And the first system that we are going to talk about is going to be the systems of respiration. And that refers specifically to the systems which utilize oxygen. And you're going to hear this term over and over and over again. Where I need to think, what is the function of oxygen? Why do we breathe oxygen, for example, ourselves? Why do most organisms require oxygen in order to respire? Because oxygen is the terminal electron acceptor. So just for a start, remember what I've said. We've got a fuel source. We grab hydrogens, whole hydrogens, from that fuel source. We strip them off, and then we're going to shunt them through a whole lot of different complex metabolic pathways and processes. And what we're doing is we're harvesting the energy of electrons. But at the end of the whole process, we cannot just release electrons. It doesn't work that way. They have to go somewhere. We need ultimately a sink for the electrons. They need to have a final resting place. And that final resting place is, in fact, oxygen. That's why we need oxygen. It is the terminal electron acceptor. And if we add hydrogens to oxygen, what do we end up with? We end up with water, or water. That's what oxygen plus 2 hydrogens is H2O. That's why water is a byproduct of these processes, because oxygen is the terminal electron acceptor. And in fact, its importance is even greater than that, because remember that when we take electrons and we put them through redox reactions, a substance is always oxidized by the next substance, which is more electron negative, that is more attractive to electrons than the substance that is oxidized. So when we think about it, through all of these complex reactions, sets of redox reactions that we're going to talk about, the last substance, the terminal electron acceptor, must be the most electron negative of all. And that is oxygen. It is extremely electron negative. And it effectively pulls the electrons towards it through all of these reactions until it ultimately accepts the hydrogens and becomes the terminal electron acceptor. And the transport of electrons stops right there. Okay, so the process of respiration refers specifically to the utilization of oxygen as the terminal electron acceptor. But this aerobic process moves through a whole set of reactions which do not themselves require oxygen. The first is a set of phosphorylation reactions called glycolysis. Glycolysis literally means breaking of sugar. The glycolysis is going to break, remember we're using glucose as our example. So what glycolysis does, that is the first step, the first set of reactions, and they are phosphorylation reactions. And it produces a small amount of ATP, not very much, as you'll hear. But it produces an end product. Glycolysis produces the end product of pyruvic acid. In fact, produces 2 molecules, which are able then to enter the next stage of reactions. There's a little preparatory step here, by the way, and called the intermediate step, sorry. They then enter a series of metabolic reactions which form a cycle. A cycle regenerates its initial substrate at the end of the cycle. Look what's happening. We're not talking about the detail at the moment. Here is glycolysis. Glycolysis does not utilize oxygen. It's an anaerobic process. It produces a small amount of ATP, but its main product in actual fact is NADH, our electron carrier. Right there is the hydrogen, one of the hydrogens that we strip off from the fuel. Here is the Krebs cycle, which is the next stage. Look what it produces. Small amount of ATP, not much, but lots of electron carriers. And all of these electrons are transported now to a complex set of redox reactions, which are in an electron transport chain. And this is what produces the bulk of the ATP in all of these reactions. The most important products of all of these early reactions here is electron carrying, NADH and FADH2, and those electrons are transported to an electron transport chain, which very efficiently produces lots of ATP, as we'll hear. Now, if I take away oxygen from an organism, especially a microorganism, many microorganisms are able to continue metabolizing. They are able to continue producing ATP. There are a couple of strange things about this when we take away oxygen, as we'll hear, but many of them pass through a set of reactions which are referred to as fermentation reactions. And fermentation reactions, like in the yeasts and in many bacteria, fermentation reactions are able to produce ATP, a small amount of ATP through glycolysis, but it's enough. It's enough for them to continue to metabolize, but their metabolism is much lower, much slower than that of aerobic organisms, which can produce masses of ATP. We'll hear about this in detail. So these are two predominant ways of extracting energy from sugars that we are going to be talking about: respiration, dependent on oxygen, and fermentation, which is not. Okay, so let's have a look at glycolysis. My first admonition to you is don't freak out, because I do not expect you to remember every intermediate, sub-intermediate, and everything. There are a couple that I do want you to remember, but in general, if I'm going to ask you detailed questions about glycolysis, I will give you the pathway. But there are a few that you must be able to remember off the top of your head. So here we start. We're going to start with our foundation substrate, which is, in this instance, is going to be glucose. Glucose is a 6-carbon sugar. 1, 2, 3, 4, 5, 6. Now, in glycolysis, remember, glucose is a very stable molecule. It's not broken down very easily. And in glycolysis, there is a preparatory step in which we're actually going to sacrifice some ATP. What's going to happen is we're going to phosphorylate this glucose molecule. We're going to add phosphates to either end. There goes one, ATP, ADP. Where does its phosphate go? It goes here. Next one, ATP, ADP. Where does its phosphate go? It goes here. And it creates this compound here, fructose 1,6-diphosphate. And that I want you to remember. Okay, but this is an important intermediate, because what happens now is that this is enzymatically cleaved. It's very easy to cleave because it's actually really rather unstable, but it is cleaved here. And don't worry about this step. All I want you to remember is this here. This compound here is converted to this here. So just remember this. So we're going to cleave this 6-carbon into 2 molecules of 3-carbon, and that is glyceraldehyde 3-phosphate. Remember that. Put stars next to these, okay? Fructose 1,5-diphosphate, glyceraldehyde 3-phosphate. Remember, important. There are 2 molecules of this. You end up with 2 molecules of glyceraldehyde 3-phosphate. This next part, this is why it's important. This next part here gives you this total of the products from both of these molecules. Okay, so if you were just to follow one, you would see that there'd be one molecule of NADH, but there are two glycerol that had 3-phosphate. So what we end up is two molecules of this NADH. Here we go. Look at this. Now we're going to be talking about actually harvesting energy. And what is going to happen is, here is NAD plus. We're going to add the phosphates, and what we end up with is NADH. And what we've done is we're going to add a phosphate to this, and we end up with this compound here. This is what is this process here of adding a phosphate from outside? That is substrate level phosphorylation. But in this case, what we're phosphorylating is this compound here. Here's a phosphate there, here's a phosphate here. This is relatively unstable, and we're able now to phosphorylate ADP, and we end up with two molecules of ATP. Follow this down, we're going to do the same thing, and we're going to end up this compound here. It has a phosphate there. We're going to, don't worry about the intermediate processes, okay? All I want you to know is we're going to take phosphate-rich compound here, strip a phosphate to phosphorylate that to get ATP. Here we've got another compound with a phosphate on it. We're going to strip that phosphate, and we're going to produce two more molecules of ATP. What have we done? Sum total, we invested 2 molecules of ATP. We produced 4 molecules of ATP here. Our net, we gain 2 molecules of ATP. Even more important probably, we end up with 2 molecules of NADH there. And that's, watch that, because there is the energy from the fuel. Now locked, the electrons now locked into an electron carrier. At the end of it, what have we got? Sum total, we got two molecules gained of ATP, and we end up with this final compound here. We end up with 2 molecules of pyruvic acid. Remember, these are substrate level phosphorylations to produce ATP there. And we end up with completely dephosphorylated. There's no phosphates associated with pyruvic acid. It's just a simple 3-carbon compound. And now we're going to follow the fate of 2 molecules of pyruvic acid. Okay, I don't know that I need you to look at the accepting. Yeah, come back to this when you do your revisions of the whole process. Okay, come back here to work out exactly what the budget is for ATP, NADH, etc., etc., etc. Okay. So now we're going to now follow the pyruvic acid through a really complex set of reactions that are put into a special kind of metabolic pathway. And this is a cycle. It's called the Krebs cycle or the citric acid cycle. Before the pyruvic acid, however, can enter into the cycle, it has to be modified. And it is modified by stripping off, remember, these are 3 carbons, right? And it has hydrogens associated with it. And those hydrogens, where are they derived from? They derive from the original glucose. So we can strip those off and harvest the energy from them. And this is the way it's done. It is, the pyruvic acid is reacted together with this compound, which you've heard about before. It's a coenzyme called coenzyme A. And coenzyme A reacts to move, to to facilitate the reduction of NAD plus, here it is here, NADH there. And remember, we will have produced a proton as well. In the process, look what a byproduct of that reaction is. It's carbon dioxide. This is the first molecule of carbon dioxide that is produced. Breathe out. What are you breathing out? You're breathing out carbon dioxide. Part of the carbon dioxide you're breathing out comes from this very reaction here, which is called the intermediate reaction. And what we end up with here is a compound. Here's coenzyme A, right? Reacting with pyruvic acid. We end up with this really important compound called acetyl coenzyme A. Acetyl coenzyme A is important because this is the way in which this compound derived from pyruvic acid, which is derived from glucose, enters the cycle. So this is what is meant by a cycle. A cycle has a substrate, an initial substrate. In this case, it's called oxaloacetic acid. Remember the name, but even more importantly, remember it is a 4-carbon compound. 1, 2, 3, 4. How many carbons in acetyl coenzyme? Well, it's an acetyl group. It's 1, 2. Those are the carbons we're interested in, because the acetyl coenzyme A unites with this oxaloacetic acid to form this 6-carbon compound. Citric acid, 1, 2, 3, 4, 5, 6. The coenzyme A is recycled. Off it goes. It can go and do any number of reactions. But what we end up with, oxaloacetic acid, 4 plus 2, 6. Now there's going to be a whole series of reactions. What we need to do is we need to count our carbons as we go through. Here we go here. We're going to go through to this compound here, which is called alpha-ketoglutaric acid. I'm not expecting you to remember, but what I do want you to notice, here we have NAD+, the oxidized form of our electron carrier. We reduce it to NADH and a proton. In the process, we're going to strip off a carbon. What has happened to the hydrogens that were associated with that carbon? They've ended up here. That is what is used to reduce the NAD+. This carbon is stripped off. Its hydrogens are harvested here. And what do we end up with? We end up with carbon dioxide. We produce there carbon dioxide. And by the way, this oxygen, the carbon dioxide, the O2 there is not derived from oxygen that you breathe. It's in the compounds that are being oxidized. So we end up with this alpha-ketoglutaric acid. Same story. 1, 2, 3, 4, 5. We're going to strip off a carbon and all of its hydrogen. We're going to harvest the hydrogens. We're going to produce carbon dioxide there. And this, by the way, is going to be facilitated again by coenzyme A. But what's really important, we reduce the NADH, produce NADH, carbon dioxide molecule, and this substance here, which is succinyl coenzyme A. The coenzyme A came from there, plus the 4 carbons there, forms this succinyl coenzyme A. So now comes a little step. Don't worry about this. All I want you to realize is that we're going to produce a small amount of ATP here. And what we're going to end up doing is producing this substance here, which is succinic acid. What is important about this is that this is a cycle. And in a cycle, we have to regenerate this substance here, oxaloacetic acid. That is done again by oxidizing this succinic acid, producing an electron carrier, FADH2. We do it again. We oxidize this intermediate to produce NADH, and we end up with our oxaloacetic acid again, ready to unite with this acetyl group here and go through the whole cycle. This is called the Krebs cycle for the person who discovered it or elucidated it, but it's more properly referred to as the citric acid cycle, because the citric acid is the first product of the cyclic metabolic pathway. What do you need to remember? You need to remember the process of the intermediate step, which produces NADH and CO2. You need to remember that it produces acetyl coenzyme A, which unites with oxaloacetic acid to produce the first intermediate, which is citric acid. That is put through a whole series of oxidation reactions where the carbons and the hydrogens are stripped, the hydrogens donated to electron carriers, the carbons breathe out as carbon dioxide, and in the end, a small amount of ATP is produced, but masses there, and there, masses of electron carriers, carrying electrons with all of their energy. And now we're going to watch what the fate of these electron carriers is. So break this down and tell yourself a story. Oh, sorry, here's a better diagram. Break this down and tell yourself a story. Don't worry about intermediates that I have not told you to remember, okay? You can, obviously. If it makes it clearer for you to remember certain intermediates, then that's absolutely fine. But what you want to do is you want to follow the carbons and the hydrogens. Where do carbons, what number of carbons do you start off with? Remember that each of those carbons has associated hydrogens. As you move through these metabolic pathways, carbons are stripped off, their hydrogens are harvested, carbons are breathed out as carbon dioxide, and the hydrogens are utilized, bound to electron carriers, and we're going to see their fate right now. Okay, so we now, all of those processes, glycolysis, the Krebs cycle, etc., take place in the cytoplasm of the cell. Remember, we are talking about prokaryotes. By the way, a number of you need to remind yourselves about what a prokaryote is, because like 2/3 of the class were unable to answer questions about what prokaryotes were. It's very important that you understand that, because we are largely discussing prokaryotes. So the early stages take place inside the cytoplasm, but now we need to move to the cell membrane. The reason we need to move to the cell membrane is we need now to have a barrier. We need to have an insulation system where we can change conditions on one side relative to the other side, because that is how we're going to harvest the potential energy, the energy of the electrons. We're going to convert the energy of electrons into potential energy and then harvest the potential energy to produce ATP. And that is done at the membrane, at the cell membrane, because in prokaryotes, that's the only membrane. In eukaryotes, this process is accomplished at the mitochondrial membrane, inside the mitochondria. But we are not going to discuss that. That you would have discussed in biology. So what we're going to see is that bound into the membrane and associated with the membrane are a whole series of substances which are able to accept and donate electrons. So this forms an electron transport chain. When you look at the electron transport chain, it has a beginning and it has an end. And each substance down the chain is more electron negative than the substance above it. So, for example, this substance is able to abstract electrons from this substance before it. In other words, this substance oxidizes that substance, and it is self-reduced because it's accepting electrons. However, it's sitting right next to another substance, which is more electron negative than it. So this substance, the next substance, is going to be able to oxidize that and itself be reduced. Same here. This is more electron negative than that. It oxidizes that and is reduced, etc., etc., etc. And what I told you before, at the end of the whole process, we're going to have an terminal electron acceptor, a final electron acceptor, where we can sink the electrons that we've used. The really important thing about this is that as the electrons move through this electron transport chain, their quantum energy falls. And this system is designed to harvest the energy, harvest at least some of the energy that is lost from the electron. And it's done in a particular way that I will describe. But the process whereby ATP is produced is a system of chemiosmosis. That means that we remember that we're at the membrane. We are going to create a system where on one side of the membrane, we have a higher concentration of something. In fact, that higher concentration is of protons. Protons are a chemical, right? They're a hydrogen ion. So it's a chemical. If we accumulate more on one side of the membrane than the other, the tendency would be to try to equalize the concentration on both sides by diffusion. But that diffusion is by osmosis. And so what we're looking at is, first of all, the diffusion of a chemical, a hydrogen ion. But second of all, we are watching that process take place by a process of osmosis. So we are going to call it chemiosmosis. It is the movement of both charge and substance down a concentration gradient. And when we do that, it's going to release energy, and we are going to harvest that energy to produce ATP. Sounds complicated. Well, I guess it is, but it'll be more clear when I've explained this particular diagram to you. So first of all, let's get ourselves oriented. This here on the outside, this is the outside. This is the periplasmic space of a gram negative, or just the outside of a gram positive. And in the eukaryotes, it's in the mitochondria. We're not going to worry about that. But notice that it's the same kind of system, right? Why? Because the system evolved in the prokaryotes. So here are the substances in the electron transport chain. Here they are here. No, they're complex substances lined up, geographically located near one another. And remember, each one down the chain, more electron negative than its neighbor up the chain. So the very first thing to notice is that this substance, the first one here, is able to accept hydrogens from electron carriers. So here where this is NADH, and remember, it always has an associated proton. Here it comes. Where did that come from, the NADH? Well, it came from the processes that we've outlined before. It came from glycolysis, some of it. It came from all of the processes associated with the citric acid cycle. And this was the purpose of making it, so that the electrons could be transported to the electron transport chain. This is a powerfully electron negative substance. It's not as powerfully electron negative as that, but it's powerfully electron negative enough that it can be reduced, it can oxidize NADH. That is its first purpose. It grabs electrons from the NADH. It oxidizes NADH to NAD+, but it has another purpose, and that is it is also a proton pump. Remember, associated with NADH is a proton. They're protons, by the way, all in solution all over the place here, but this substance is able to pump protons out to the other side of the membrane. But remember, this substance here is more electron negative than that. This substance is able to oxidize this. It grabs the electron and is itself reduced. This substance is also important because it accepts hydrogens from FADH2 there. This is not a proton pump, by the way. But it can oxidize FADH2, grab the hydrogens and the electron, grab the electrons rather, and what does it leave behind? It leaves behind protons. This is FAD plus 2 protons. The next substance is more electron negative than that. It is able to oxidize that, grab the electrons, and it is itself a proton pump. It pumps protons out. Where's it getting the energy to do that? It's getting the energy from the loss of energy by the electrons as they are grabbed by the next electron negative. As the electrons come down through this chain, they gradually lose energy, and that energy is harvested by the proton pumps to pump protons to the other side of the membrane. Same story here. Here's another proton pump. It's going to pump protons out. So what have we done? These three substances, all of these substances here are harvesting electrons from electron carriers. As they stream down this electron transport chain, the electrons lose energy. The energy is harvested by the proton pumps, and they pump protons out to the other side here. So what is happening? We've got all sorts of differentials being built up. First of all, this is a chemical. It's a hydrogen ion. There are more hydrogen ions on this side than there are on this side. Second of all, this is carries a positive charge. This is much more positively charged on that side of the membrane than the relatively negative charge that is accumulated here on this side. Thirdly, accumulation of protons means that this is acid. This is more acid than this side. We've got all these sorts of differentials being built up across the membrane. Just think of the one that I have explained before. If I have an insulating barrier like a membrane, and I accumulate charge on one side, so there's a charge differential, what do I have? I have a battery. That is how a battery works. A battery works because it accumulates potential energy. And we have three sources of potential energy here. First of all, the substance itself, the hydrogen ion. Second of all, the charge the hydrogen ion carries. Thirdly, the fact that this is more acid than on this side. The cell now can harvest that. It can harvest this is the equivalent of putting a wire from one pole of a battery to the other. Because we've done all of this, we've accumulated all this charge, now we need to use it. Before we do, we need to think though. We have electrons streaming through here. they have to go somewhere. They have to be accepted by a strongly electron negative substance. And the most powerful electron negative substance in this whole system is an atom of oxygen. And the atom of oxygen accepts ultimately, and the reaction is a little bit complicated, but you don't need to worry about it. We end up effectively, each atom of oxygen accepts 2 hydrogens plus their electrons. So it accepts 2 protons plus 2 electrons and forms molecules of water. And that is why you breathe out water. You breathe out carbon dioxide from the preceding metabolic pathways, the intermediate step and the Krebs cycle. You breathe out water predominantly from these reactions here, as oxygen acts as the terminal electron acceptor. So how are we going to harvest the potential energy? We're going to turn to this incredible molecule here. I won't discuss it in detail, but it is called ATP synthase. Here's the name here. This is ATP synthase. If it's a synthase, it's going to be manufacturing. It's going to be manufacturing ATP from ADP and phosphate. And in order to do that, it's going to require energy. And the energy is derived from the flow of protons, because this is not only an enzyme which will phosphorylate ADP, it is also an enzyme which is a transport protein. And it specifically transports protons from the outside of the membrane, accumulated there by all of these pumps. They flow through the ATP synthase. And as they do so, the ATP synthase harvests their energy in order to phosphorylate ADP and to produce ATP. In the process, by the way, it's allowing now these protons now on this side again to unite with electrons and oxygen to form water. So that is basically the whole system. And I know that it's complex, and it's probably difficult to grasp at first go round. But what I want you to do is to go through the systems really carefully and draw up a budget. Draw up budgets for each of the substances. Draw up a budget for NADH, for example, for FADH2, for ATP, for CO2, and for water. And see where those molecules are all produced. And follow, where do the carbons get stripped off? Where do the hydrogens get stripped off and put into the electron carriers, etc. And as you do that, it'll start to become more clear. So here is a useful budget for you, which you can use as a basis for your own budgets. But I don't want you to just copy this. I want you to actually go through those metabolic pathways and find for yourself where these are derived from. And in the end, this is the commonly accepted amount of ATP which is produced from one molecule of glucose. Here's where the sources of the ATPs, however, how many we derive from, for example, from the NADH that's produced during glycolysis, how many are produced from the substrate phosphorylation, etc., for each stage. And in the end, 32 ATPs from one molecule of glucose. That is the accepted budget for aerobic respiration. Now, I say that's the accepted. In fact, there is some variability. One of the major sources of variability, which I'll tell you about when you see class from Tuesday. One of the sources of variability is cells don't always use glycolysis at the beginning. That's one. Second of all, all of these processes from glycolysis and citric acid cycle produce very, very useful intermediates. And those intermediates are often spun off into other metabolic pathways. And when they reduce the amount of ATP that is finally produced. So there is variability in the amount of ATP produced from one molecule of glucose. But all things being equal, we accept 30. If I ever ask you how many are produced, 32 is a good answer. Okay. So I'm going to leave it there for the day, and then I'll complete the rest in our class on Tuesday. Okay.